Welcome to the Explainer. Today, we're diving into something called AntiFold. It's an AI model that is completely changing the game when it comes to designing antibody drugs. This is all about teaching a machine to think like a molecular architect, so we can build better, life-saving medicines way faster than ever before. So let me ask you this. Imagine you're trying to upgrade a race car's engine while it's in the middle of a race, right? You have to make these super precise tweaks to get more performance, but just one wrong move, one tiny mistake, and the whole thing could just catastrophically fail. Well, that's pretty much the high-stakes world of engineering therapeutic antibodies. You know, antibodies are a huge deal in medicine. They're one of the most successful classes of drugs we've ever had. But actually designing them from the ground up, it's like solving an incredibly complex three-dimensional puzzle. Scientists are constantly trying to balance everything at once. You need the drug to be effective, it has to be safe, and it needs to be stable enough to actually be made into a medicine. And here's the thing you absolutely have to understand. An antibody gets all of its power from its specific 3D shape. Its structure is its function. So if you make even a tiny little change to its amino acid sequence, maybe to make it bind to a virus better, you could accidentally shatter that crucial 3D structure. And just like that, the drug is completely useless. So this huge challenge got scientists thinking, what if we've been looking at this problem completely backwards? Instead of starting with the sequence and just, hoping it folds the right way, what if we could start with the perfect 3D structure and then work our way backward to find a sequence that builds it? This is the core idea that makes AntiFold so incredibly powerful. This whole concept is called inverse folding. And honestly, the best way to think about it is like this. Imagine you hand an architect a perfect blueprint of a finished skyscraper, and you ask them for a list of all the possible materials that could have been used to build that exact structure. That's what we're doing here, but for proteins.
So how does AntiFold actually work day to day? Well, the workflow is actually beautiful in its simplicity. First, you give it the 3D structure of an antibody you want to improve. Then the model gets to work. It looks at every single position and calculates the odds of each of the 20 different amino acids working in that spot. And what you get at the end is this amazing menu of new, diverse sequences that are all predicted to fold into that same perfect target shape.
Okay, so how on earth did the researchers build an AI smart enough to master such a specialized job? This is where we get to peek under the hood and see the really clever engineering that makes AntiFold tick. Right, so you can't just use a generic, off-the-shelf protein model for this job. Antibodies are, well, they're special. The parts that do all the important work, the bits that actually bind to a target like a virus, are called CDR loops. And these loops, especially one called CDRH3, are just wildly diverse. Their shapes and structures are unlike almost anything else in the entire protein universe. A general model just doesn't have the training to get those unique parts right.
And hey, they have the data to prove it. When the researchers first tried to build a model trained only on antibodies from scratch, the results were not great. Its accuracy at predicting the right amino acids for that critical CDRH3 loop was a measly 31.5%. That was a total non-starter and a clear sign they needed a much, much smarter approach. So the big breakthrough, the real game changer, was deciding not to start from scratch. Instead, they built AntiFold on top of a massive existing model called ESM-IF1. You can think of it like this. They started with a student who already had a PhD in the general physics of proteins, since ESM-IF1 had already learned the fundamental rules from studying over 12 million different protein structures. Then they took this genius model and put it through a specialized training program just on antibodies. And they used a really clever two-part strategy. They fed it over 2,000 high-quality, real-world structures to learn the ground truth. But they also gave it this massive library of almost 150,000 predicted structures. This gave the model the sheer volume it needed to learn the incredible diversity of possible antibody shapes. Now, this is where it gets really fascinating. They didn't get to the final version overnight. They built up its performance piece by piece. They started with a basic technique where they'd hide a chunk of the protein and make the AI guess what's missing. That got them to about 36% accuracy. Then they added something called shotgun masking, which is like hiding random bits all over the protein. This forced the AI to learn the global context of the whole shape, and boom, that jumped the accuracy up to 53%. After a few more clever tweaks, the final massive leap to over 58% came when they added that huge data set of predicted structures we just talked about. All right, so that's a lot of complex training, and it sounds really impressive on paper. But did it actually work? Let's take a look at the numbers and see how AntiFold stacks up against the competition.
Okay, first up, let's talk accuracy. This is a big one. It basically asks, how often does the model correctly predict the right amino acid for the most important part of the antibody, that CDR-H3 loop? And the results are crystal clear. At 60% accuracy, AntiFold is the decisive winner, easily beating its closest competitor and leaving the original general model in the dust.
But being accurate is only half the battle. The real test is whether the new sequences the AI designs will actually fold into the correct shape in a real-world lab. This chart measures that, and here a lower number is better. And once again, sequences designed by AntiFold show the highest structural fidelity. That means they're the most likely to actually work, which saves a ton of time and money on failed experiments. So if we just pull back and look at the overall scorecard, the story is incredibly consistent. It doesn't matter what metric you look at, from predicting the sequence to making sure the final structure is right, to even predicting how well it will bind to its target. AntiFold is consistently outperforming all the other state-of-the-art models.
So, these technical results are obviously fantastic, but let's talk about what this all means in the real world. Why does this kind of superior performance actually matter when it comes to developing new medicines? Well, this right here is the real payoff. A tool like AntiFold can speed up pretty much every single stage of antibody design. It helps scientists make a drug bind more tightly, it streamlines the process of making sure a human immune system won't reject it, and it helps them design better, more focused experiments. At the end of the day, it's about doing smarter science, pre-screening designs on a computer to radically cut down on expensive, time-consuming work in the lab. The researchers who built it really summarized it perfectly. They said that tools like AntiFold represent a true paradigm shift. We're moving away from a slow trial and error process and into a new era of a structure-first, computationally guided cycle of design, test, and learn. It's an absolutely incredible advance, and it leaves us with this one big provocative thought. If we have AI that can understand and design the fundamental building blocks of medicine with this level of precision, are we getting close to an era where we can design the perfect antibody for almost any disease, basically on demand? The journey is really just beginning.